The overall goal of the following experiment is to photoactivate a given light-responsive agent, such as chemically caged fluorescent dye, in a live zebrafish embryo at a single-cell resolution. This is achieved by injecting a light-responsive agent into one-cell stage embryos expressing a live genetic landmark to locate and precisely target any cells of interest. As a second step, embryos are grown and mounted in a low melting point agarose, which immobilizes the live embryo. Next, two-photon microscopy is used in order to localize a visible fluorescence transgenic landmark. The light-responsive agent is then activated in a desired focal plane, and the extent of photoactivation is monitored. The fate of the resulting labeled cells can then be traced at later embryonic stages. This procedure can be utilized for the activation of a variety of compounds at a single-cell resolution in live specimens.
The main advantage of this technique over existing methods such as confocal microscopy or flash lamp is that it allows photoactivation of relatively deep tissues at axial level of a few microns with relatively low phototoxicity.
The method presented here is used for cell lineage tracing. It can be applied for photoactivation of a variety of compounds, and that's allowing targeted control over developmental and physiological processes.
On the evening before the injection, prepare mating tanks. Separate male and female zebrafish of a transgenic line expressing a visible fluorescent landmark. In this experiment, the transgenic line expresses GFP under the control of neurogenin-1 promoter. Thaw an aliquot of 5% dextran conjugated caged fluorescein in